My name is Zainal Shah and I am from Group 8 and Semester 1. Today my topic is natural antigens and human antibodies and history and discovery of blood. The human ABO blood group was discovered by Austrian-born American biologist Carl Landstrom in 1901. Landstrom found that there are substances in the blood, antigens and antibodies that induce clumping of red blood cells when red cells of one types are added to those of second types. And the second one is the types of natural antigens and ABO blood system. There are four types of ABO phenotypes are O, A and B. And AB, after it was found that blood group A or red blood cells reacted directly to particular antibodies. Later anti-A and the blood group was divided into two phenotypes A1 and A2. The red blood cells with the A phenotypes react with A1 and the blood group which A2 phenotype react with A2 and 80% of blood types. The ABO system blood group A has of antigen on the red blood cell with anti-B and antibodies in the plasma. Blood group B has B antigens with anti-A antibodies in the plasma. Blood group has no antigens but both antibodies in the plasma. There is a third one is the types of natural antibodies. The human antibodies are classified into 5 isotopes: IgM, IgD, IgG and IgA and IgE according to their H chain, which provide each isotopes to distinct characteristics and role. And the fourth one is geographical distribution of blood group. Blood group provide an ideal opportunity for the study of human variation without culture project. This can be classified for many different genetically inherited blood typing system. Also significant is the fact we are rarely take blood taps into the consideration in the selecting mates. Now the blood transfusion, it is the process of transfusion blood product into a person circulating intravenously, and transfusion are the used for various medical conditions to replace lost components of the blood. Early transfusion used whole blood, but modern medical practice commonly use only component of the blood, such as red blood cell, white blood cell, plasma, clotting factors, and platelet. Red blood cells contain hemoglobin and supply the cell of the body with oxygen. White blood cells are not commonly used during the transfusion, but they are part of the immune system and also fight injection plasma in the yellowish liquid part of the blood, which act as a buffer and contain protein and important substance needed. And for the body overall health, platelets are involved in the clotting, preventing the body from bleeding. The fifth one is the blood transfusion, and the blood transfusion are generally considered safe, but there is some risk of complication. Mid-complication are rarely solved when a cause occurs. During the transfusion of several days or more after, more common reaction include allergic reaction, which might cause hives and itching and fever. But the allergies caused by blood transfusion error can include. That's one is the infection, hepatitis B, hepatitis HIV, acute hemolytic transfusion reactions. When a patient antibodies destroy donor blood cells, leading to the antibiotic shocks, stroke, blood clotting, or kidney failure.